Today, we're comparing two fundamental processes, replication and transcription. Both involve nucleic acid synthesis using templates, but they differ in mechanisms, accuracy, and outcomes. Let's break it down. Transcription synthesizes RNA, like mRNA, tRNA, or rRNA, using a DNA template. The information remains in the same language, a sequence of nucleotides. Transcription transfers genetic information from DNA to RNA. Replication and transcription share key similarities. Both rely on template-driven polymerization. In replication, DNA polymerase synthesizes a complementary strand of DNA using the parental strand as a template. Similarly, in transcription, RNA polymerase synthesizes RNA by reading the DNA template strand. In both processes, the driving force is the removal and hydrolysis of pyrophosphate from each nucleotide added to the growing chain. This makes the polymerization process thermodynamically favorable. Replication and transcription both proceed in the 5-3 direction, meaning new nucleotides are always added to the 3 end of the growing strand. The directionality ensures that the polymerase enzymes can work efficiently and sequentially along the template. Both processes begin at specific sites on the DNA. Replication starts at the origin of replication, a sequence where DNA polymerase and other enzymes assemble to begin synthesizing the new strands. Transcription begins at the promoter, a sequence that activates RNA polymerase to initiate RNA synthesis. The actual site where RNA polymerization begins is called the start site. The enzymes driving these processes are different. In replication, DNA polymerase synthesizes the new DNA strands. In transcription, RNA polymerase synthesizes RNA. Another critical difference lies in their proofreading abilities. DNA polymerase has exonuclease activity, meaning it can remove and correct mismatched nucleotides, ensuring high fidelity during replication. In contrast, RNA polymerase lacks exonuclease activity, making transcription a lower fidelity process. Errors in transcription are less critical since RNA is temporary and not passed on to offspring. The ultimate goal of these processes is different. Replication produces an exact copy of the entire DNA genome, ensuring genetic information is preserved for cell division. Transcription produces a single-stranded RNA molecule, which serves as a functional intermediate, either coding for proteins or regulating gene expression. Step-by-step comparison. Replication, initiation. Begins at the origin of replication. Elongation. DNA polymerase adds nucleotides to the new DNA strand in the 5-3 direction. Termination occurs when the entire DNA molecule is replicated. Transcription. Initiation starts at the promoter, where RNA polymerase binds and unwinds the DNA. Elongation. RNA polymerase synthesizes RNA by adding ribonucleotides complementary to the DNA template. Termination ends at a termination signal, releasing the RNA transcript. Test your knowledge. Which of the following is true about transcription, but not replication? A. It uses RNA polymerase. B. It occurs in the 5-3 direction. C. It relies on a DNA template. D. It has proofreading activity. Pause here to think about your answer. The correct answer is A. It uses RNA polymerase. While both processes rely on template-driven synthesis and occur in the 5-3 direction, only transcription uses RNA polymerase. Errors in replication and transcription have different consequences. Errors in replication can lead to permanent mutations in the DNA, which may cause genetic disorders or cancer. Errors in transcription affect only the RNA and the proteins produced from it. These errors are temporary and do not affect future generations of cells. Applications in biotechnology. In biotechnology, both replication and transcription are harnessed for applications like PCR, DNA sequencing, and RNA vaccine production. Understanding their mechanisms is essential for these advancements. Let's summarize. Both replication and transcription rely on template-driven polymerization and occur in the 5-3 direction. Replication uses DNA polymerase, requires a primer, and has proofreading activity, ensuring high fidelity. Transcription uses RNA polymerase, does not require a primer, and is less accurate but more flexible. The final products differ. Replication produces double-stranded DNA, while transcription produces single-stranded RNA. Want to deepen your understanding of replication and transcription? Visit kotc.me for interactive quizzes and detailed resources to test your knowledge. Thanks for watching, and see you next time.